Schön, dass Sie wieder dabei sind oder sich neu dazugeschaltet haben. Herzlich willkommen im Namen der Jung-Stiftung für Wissenschaft und Forschung in Hamburg zu unserem zweiten Teil des Ernst Jung-Symposiums für medizinische Forschung 2021. Nun kommen wir zum letzten Vortrag dieser Veranstaltung. Es ist unser Preisträger der Ernst Jung-Medaille in Gold. Er wird nun seine herausragenden Forschungsergebnisse ebenso wie die anderen im Livestream präsentieren. Und ich begrüße in unserem Studio Herrn Professor Büchel und Herrn Professor Brüning. Herr Professor Brüning, Sie führen uns netterweise durch den Vortrag.
Thank you very much. It's a great pleasure to announce to you the final highlight of this Ernst Jung-Symposium, presented by this year's recipient of the Ernst Jung Gold Medal, Antonia Lanzavecchia. And it's a great pleasure to briefly introduce him to you, although probably most all of you know him really well. So Antonio Lanzavecchia studied medicine at the University of Pavia. He then specialized in pediatrics and infectious diseases. Subsequently, he worked at the University of Genoa, then at the Basel Institute of Immunology, and from 1999 to 2020 as the founding director of the Institute for Research in Biomedicine in Bellinzona. Recently, he took on a new position at the Istituto Nazionale di Genetica Molecolare in Milano, where he continues his research. In light of his seminal, groundbreaking discoveries in the field of immunology, he already received several prestigious awards, including the EMBO Gold Medal, the Cloetta Prize, the Robert Koch Prize, and the Louis Jeantet Prize for Medicine, among many, many others. And as for the other speakers, we're very much looking forward to a brief video portrait of Andrea Lanzavecchia.
My name is Antonio Lanzavecchia. I'm an immunologist. My interest is to study the human immune system, how our cells react to pathogens in order to find new antibodies and new therapies. And after working for 40 years in Switzerland, I've just moved to Milano to start a new laboratory. I was trained as a pediatrician. I was studying infectious diseases. And so I became interested in understanding the mechanisms that protect us from infectious diseases, the basis for vaccination and other immunotherapies. And so at the time, immunology was not advanced as it is now. And so I thought it would have been important to develop some reliable and robust method to study the immune system. So I started to culture the human cells in vitro. I was able to clone these cells to study their specificity and to study the way they interact. One of the important milestones was the discovery of a method to readily isolate monoclonal antibodies from memory cells. And using this method, we have developed antibodies that are now used to treat Ebola virus infection. You know, they can save lives of individuals which are already infected by the virus. I have to confess that I feel myself very lucky and privileged to have such a motivation from my own work. You know, coming to work in the morning, looking at new data as they come out is a joy for me and really keeps me alive. When I don't work, I like to hike in the mountain, a little bit of skiing, which brings me back to what is most essential of life. And when I'm not able to go to the mountain, yeah, even walking in a city is beautiful. And this helps me organize my thoughts and think maybe of the next experiment. So I'm very humbled and privileged to receive the Ernst Jung Medal for Medicine in gold. And I like to think that this could be an example for young doctors, as I was at the time, that want to understand better the mechanism of protection from infectious diseases. We don't know what the future is going to reserve us, but we have very interesting topics to study. We have new technologies, and I'm sure that we are going to get a lot of exciting findings in the next several years. I'm really looking forward to that.
Dear Professor Lanzavecchia, please also accept my very personal congratulations on this very recent award to you. And we're very much looking forward to this, in these days, more timely than ever, presentation on antibodies and vaccines, the immune systems at work. We're very much looking forward to your presentation.
Thank you. Let me start by thanking the Curatorium and the Jung-Stiftung for this very prestigious prize. I'm really honored and humbled. In the next 25 minutes, I will go through our experience of isolating antibodies to try to develop them for serotherapy, but also to learn how to make better vaccines. And I want to start with a golden age of German science at the beginning of last century, when many scientists, especially Emil von Behring and Paul Ehrlich, really pioneered the use of antibodies for serotherapy. The antibodies were made in horses and saved thousands of lives because, of patients with tetanus, with diphtheria, and so on. Now, this treatment is still continues up to date. In fact, for diphtheria, we still have only the horse antiserum as treatment. So the point here is that from the origin, it took really a long time until the beginning of the 2000s to develop new approaches to the use of antibodies for serotherapy. So that today there are several antibodies which are used for prophylaxis and therapy of many infectious diseases, including COVID-19. Now, in 2004, at the time of the first SARS emergence of SARS coronavirus, we developed a facile method to isolate monoclonal antibodies by immortalizing memory B cells or keeping single plasma cells alive and screening the antibodies produced by these cells. So the antibodies could therefore be selected for potency and for binding, for instance, to conserved epitopes, what we call the breadth of antibody binding. And these antibodies can be used for prophylaxis and therapy of infectious diseases, but also, as I will elaborate, can be used for designing better vaccines. So the first obvious idea was to quickly develop antibodies to counter emerging viruses. This is a scheme. So from survivor, we could isolate the B cells, immortalize, produce the recombinant antibodies to be tested in an animal model and eventually developed in humans. And here I'm just going through a few publications where we isolated antibodies to SARS coronavirus, to MERS, and to Ebola. And we were quite pleased that through this collaboration with the NIH, our monoclonal antibody 114, which is now called ansuvimab, has been tested and shown to cure Ebola-infected patients. When given early, the mortality is reduced by more than 90%. So this was quite an achievement. I'm sure you remember the threat that Ebola imposed on our society. But here is the other idea. So not only we can develop the antibodies, if you like, as a drug, but we can use the potent neutralizing antibodies as a tool to identify, if you like, the Achilles heel of the pathogen, and so then to produce a vaccine that would be able to induce the same type of antibodies. At the same time, you could study and identify the cellular receptor of the pathogen. And the first example that we had has to do with cytomegalovirus. So we identified very potent neutralizing antibodies that neutralize cytomegalovirus and found, surprisingly, that they don't bind to the fusion protein, but rather bind to this pentameric complex that determines the protropism of the virus for different type of cells. So then what we made a recombinant pentamer as a vaccine, and we found that in mice, this vaccine can induce neutralizing titers that are at least 300-fold higher than those found in CMV-infected patients. So by carefully choosing the right component of the pathogen, one can generate vaccines that are much more, confer a much better protection as compared to the natural infection. So we went on and together with other colleagues, several laboratories, including our own, found broadly neutralizing antibodies that identify a conserved region in the stem of influenza hemagglutinin. We were the first to find an antibody that neutralizes all influenza viruses. And besides the potential use of these antibodies for prophylaxis of influenza, I think these data paved the way to the development of a universal influenza vaccine that would induce the same type of antibodies. And in another study, we found that the broadly neutralizing antibodies to conserved site of prefusion F proteins in respiratory syncytial virus and metapneumovirus. And these data provide a rationale for developing a stabilized prefusion trimer as a vaccine. You see here an example of a nanoparticle, a synthetic nanoparticle that expresses a stabilized form of the prefusion protein. And finally, along the same line, the sotrovimab is an antibody that recognizes a conserved site on SARS-CoV-2 receptor binding domain. You see that the receptor binding domain is now accumulating several mutations on the so-called receptor binding site where ACE2 is bound. Now, this antibody instead recognizes a very conserved site, and so it's not sensitive to the mutations that now are accumulating on the RBD. And in fact, in a recent clinical trial that was halted for overwhelming efficacy, this antibody was found to be 85% effective in preventing hospitalization or death in individuals at risk. So I think these are examples that illustrate the general idea of going for antibodies that have a function and that recognize conserved sites on the pathogen. Now, I want now to change gear and tell you about this analysis applied to the immune response to Plasmodium falciparum. And this is really, if you like, a fight between two evolutionary titans: the parasite that has a large number of very polymorphic genes and the immune system. So who will win at the end? Now, the parasite has very smart strategies. And the first strategy is that the infection is a kind of stealth infection. The infecting agent is the sporozoite, which is introduced in minute amounts by the mosquito. Just 20 to 200 sporozoite travel quickly through the blood and home to the liver, where they develop into the blood stage. Now, the sporozoite surface protein, which is called CSP, is conserved and does not induce an effective antibody response in infected individuals, probably because there is so little of this protein introduced by the mosquito at any time point. So the idea is to develop a CSP-based vaccine that could confer sterilizing immunity by just blocking the parasite at the entrance. But the current vaccine, which is called RTSS, is not very effective. Efficacy ranges from 30 to 50%. So the question was, can we improve the CSP-based vaccine? So as part of the very fundamental studies on the mechanism of protection, we were able to obtain blood from volunteers that were immunized with large amounts of irradiated sporozoites. These individuals made antibodies and were protected from the infection. And when we isolated antibodies from these protected individuals, we found that the antibodies have unusual properties. They bind to multiple sites on the CSP protein that you see here. And these sites comprise not only a region which was well known, which is called the NA-NP repeat region, but also and importantly bind to N-terminal peptides, which we call NPDP or NVDP. And this dual capacity to bind to these two sites is essential for the highly protective potency of these antibodies. If you see here on the right, the current vaccine does not contain this N-terminal region, so these antibodies could be used not only to confer passive protection, but also suggest that the malaria vaccine could be improved by including in the vaccine this N-terminal peptide in order that so that this type of antibodies could be induced. Now, I want to change to the so-called blood stage parasites. And here, the situation is completely different. There are something like 10 to the 11 blood stage parasites. So there is a huge antigenic load in the circulation. But here, the parasites uses a different strategy. I like to say it's a master of disguise, can continuously change its antigenic surface. So the blood stage parasites express these variant surface antigens on the surface of infected erythrocytes. These mediate adhesion of the infected erythrocytes to the endothelia. We know that anti-VSA antibodies can protect from the disease, not from the infection, but from the disease. But the parasites has about 200 genes to play with. These genes are highly polymorphic and they are clonally expressed. So if the individual makes an antibody for the parasites, it's very easy to change the variant surface antigen. So the question we had was, can we find antibodies that broadly recognize these infected erythrocytes? And so we started by screening, very simply by screening plasma from approximately 500 individuals that live in Kenya and are exposed to the malaria parasites. And we identified 3 donors that have plasma that can agglutinate several different parasites that you see here are labeled with different colors. So these below are the isolates. These are parasite isolates within Kenya, which are genetically very different from each other. And these 3 donors have antibodies in the serum that can cross-agglutinate all or most of these parasites. So we immortalized these cells and isolated, you see here on the left, a large number of monoclonal antibodies that broadly react with most, if not all, of these parasites with different patterns, but they are very broad. these are obviously much broader than the last 3 antibodies down here that you see are very specific for a single isolate. This is what we were expected to find, and surprisingly, in these donors, we find the broadly reactive antibodies. Now, when we sequenced the V genes from these antibodies, we found something totally unexpected and surprising. We found that all the broadly reactive antibodies carry a mutated layer 1 exon, which is inserted between the V and the DJ. So normal antibodies are made by rearranging V, D, and J segments. But this insertion of a new domain was totally unexpected. In fact, the layer 1 is a collagen binding inhibitory receptor which is encoded in on another chromosome, on chromosome 19, while immunoglobulin heavy chain genes are on chromosome 14. And so if this layer 1 exon that encodes for a domain is inserted between the V and the DJ on the protein, this domain is appended on the tip of the long CDR3. And the CDR3 is known to be highly permissive for insertions because CDR3 can be, of course, of different lengths. Now, we then studied these antibodies more in details. First of all, we identified the target molecules. These layer 1 containing antibodies bind to certain refins, which are a type of variant surface antigens, and they opsonize infected erythrocytes, so they have potentially they can protect from the disease. Second point, the layer 1 domain alone is sufficient for binding to the refins. So we made recombinant proteins. The rest of the antibodies was not necessary, just the layer 1 is binding. In each individual, we isolated many different clones, but all the clones belong to a single expanded family, which is highly mutated. You see that down here in this clonal family, there are 48 amino acid substitution in the V and layer 1 gene. And finally, the somatic mutations have 2 functions. Remember that layer 1 binds collagen, so potentially is an autoantibody, but through mutations, the binding to collagen is lost. And this is an example of clonal redemptions. Mutations can turn an autoantibody into a useful antibody without self-reactivity. So this was quite an extraordinary finding, but the next question was whether this was a very rare finding and how common are these layer 1 containing antibodies. So to address this question, we systematically examined 2 other cohorts in Tanzania and Mali and found that approximately 10% of the individuals have in the serum IgG antibodies that contain layer 1 antigenic material. And what was interesting, when we cloned out these cells from these individuals, we found that the layer 1 antibodies really dominate the response to infected erythrocytes. Virtually all the anti-infected erythrocytes antibodies in these individuals contain the layer 1. So this is, again, a very unexpected findings, a case where a single clone of B cells can mount a very large and effective antibody response to a pathogen, just a single cell. So because we immortalize the B cells, we can look not only at the cDNA, but also at the genomic DNA. And for each individual, we found that distinct insertions that encode for the layer 1 exon with a little bit of intronic flanking sequences that sometimes are spliced out, this both in Kenyan donors and Tanzanian donors. And so in all these cases, the extra domain of layer 1 will be appended on the tip of the CDR3. But by sequencing other B cells, we found, again, a surprising finding that in some donors, the layer 1 exon and intronic sequences was inserted into the switch region. And because intronic sequences conserve the splice sites, these particular exon could be spliced in very efficiently. And so in this case, the layer 1 domain will be inserted after the V, between the V and the CH1. So I should say in the elbow of the antibody, which is a location where, it was not supposed to be modified or to be engineered. I think in this case, nature has done it before us. Nature always does something before humans. But anyway, so this was a surprising finding. And so the question then was whether there are other examples of templated inserts into immunoglobulin genes. Is this a rare event maybe caused by the malaria infection, or is this a general phenomenon that leads to antibody diversification? And so to ask this question, we took two approaches. The first is an unbiased analysis of the switch regions and also of the VDJ to see whether there are templated inserts. And the answer is very clear. Switch regions contain multiple inserts of different lengths. These inserts are derived from genic regions, mostly introns and exons, and in some cases, whole exons which can be spliced in. And these fragments, these templated inserts, derive essentially for all chromosomes and are inserted into antibody genes. So this work was done in European donors, so we exclude the possibility that the parasite induces in some way this phenomenon. This phenomenon is a spontaneous phenomenon, and what the parasite does is to select those rare clones where a useful insertion has been generated. The second approach was to make what we call an educated guess, and we thought, well, if the parasite targets layer 1, which is an inhibitory receptor, it may target other inhibitory receptor. And so we searched for antibodies that would contain LILRB1, which is another inhibitory receptor encoded in the same locus. And in this Malian cohort, we identified a few individuals that have LILRB1 containing IgG. These IgG stain the parasites. We sequence the genomic DNA, and we find large insertions in the switch region that comprise different exons that encode domain 3 and 4, or in one case, domain 3 only of LILRB1. And again, by differential proteomics analysis, we identify the target molecule as certain refins once again. So this is, again, an interesting situation where both layer 1 and LILRB1 are inserted into immunoglobulin genes and are selected because they bind to refin. So we search for, we try to map the binding of layer 1 and LILRB1 to the 200 refins which are present in the genome of the parasites. And what is interesting is that there are multiple refins that bind this inhibitory receptor, as you see here, that are belong to different phylogenetic groups. So the parasites employed multiple genes to bind to the same inhibitory receptor. And this means that for the parasite, it's very important to inhibit the immune response. So again, the parasites have learned this much before we did, the importance of targeting inhibitory receptors. Now, the escape from antibodies is facilitated by the clonal expression of the refins and by the polymorphism of the refins, which is extraordinarily high, as you have seen, even in the same region. And what layer 1 and LILRB1 containing antibodies do is that they recognize multiple genes irrespective of their allelic form, and then they counteract effectively the evasion strategy of the parasites. And so this is an interesting point, and I think illustrates what I was calling this clash of titans between the parasites and the immune system. And I would like to close with some structural data with this razor and cartridge model of refins binding to layer 1 and LILRB1. Here you see the domain of layer 1 and LILRB1, which are structurally similar, although the sequences are different. And here you see the refins, which look like, a little bit like a razor binding to its cartridge. And this is a layer 1 specific refins in blue and a LILRB1 specific refins in yellow. You see that the angle is slightly different, but the mode of binding is quite the same. And finally, the very unusual structure of a FAB fragment that contains the LILRB1, D3, and the D4 domain. So what you see here is an antibody where the VH and VL and the CH-CL1, CH1-CL are opened up. The hinge of the light chain can swing out about 90 degrees to accommodate the two domains of LILRB1, D3 and D4, which are inserted in the elbow between the VH and the CH1. And what is, of course, refin will bind to domain 3. And what is interesting, however, is that the pairing of VH and VL and CH1 and CL is absolutely preserved. So is preserved the specificity of the antibody, which is not affected by the insertions of LILRB1. So I like to show these slides to make 2 points. The first is that here we have a number of refins that target inhibitory receptor. This is a very interesting evasion strategy used by the parasites. There is a fundamental difference. Layer 1 is left 1 is self-reactive because it binds collagen and C1q. LILRB1 is also self-reactive because it binds HLA1 molecule. But the difference is that when these domains are inserted into the antibodies, the domain of layer 1 has to mutate away in order to avoid self-reactivity, while the two domains, D3 and D4 of LILRB1, don't have to mutate away because they are not self-reactive. Now, you see here on the right a picture of Paul Ehrlich and his famous side chain theory. Ehrlich, who was a chemist, was the first to propose a model of antibodies, and he suggested that antibodies are nothing but the side chains, so essentially the receptor present on the surface of the cells that the cells produce in an excess in order to block the binding of the pathogen. And we know now that Ehrlich's theory as such was not correct, but I would like to say that in this case, peculiar case, Ehrlich was quite right, because the antibody actually incorporates through this insertion the receptor, the essential element of the receptor. And so I like to close with this slide, where a kind of provocative title, Immunology Taught by P. falciparum. We can learn a lot by studying the immune response. We have a strategy to improve the CSP-based vaccine by by including N-terminal epitopes which are missing in the current vaccine. We found that the refins can engage inhibitory receptor and evade the antibody response through multiple polymorphic and clonally expressed genes, and vice versa, the immune system can fight back by putting these receptors into antibodies and in this way counteract this escape strategy. And at the end, we have learned how to engineer antibodies by inserting a new domain in the so-called elbow, and also how to introduce DNA fragments at double-strand breaks. So this is what happens when cells switch and the templated DNA is inserted. So with this, I want to thank you again for this great honor, and I would want to thank all my collaborators in Genova, where my career started, in Basel, where we laid the many fundamental observations, and in Bellinzona, and now in Milano. Thank you very much.
Thank you very much, Professor Lanzavecchia, for this fascinating lecture. We have one question from the audience. If you could maybe briefly comment on the mechanism underlying the integration of those foreign fragments into the VDJ locus. I mean, so what's the mechanistic basis, how those foreign genes are integrated there?
It's becoming increasingly clear that double-strand breaks, which are repaired by the so-called non-homologous end joining, can be often repaired using patches of DNA. And where does DNA come from? Well, we don't know, but possibly because of its origin, it may come from the resolution of replication forks. And this resolution involves the cutting out of pieces of DNA of approximately 1 kilobase, which are about the size of the inserts, more or less, that we find in immunoglobulin genes. Indeed, it's possible, and that's my colleague, Catherine Pieper, who is now working in Berlin, has shown that it's possible to make the B cell switch and offer an artificial substrates that then will be inserted into the double-strand break. So we can recapitulate this in vitro.
Thank you very much again. Congratulations again to the receipt of the prize. Also, same comment as for all the other outstanding lectures. Also, this one will be available online. And with that, I would like to hand back to Mr. Solde for the closing words of the symposium.
Ja, noch ein kurzer Hinweis. Erstmal auch vielen Dank, Herr Lanzavecchia, für den wunderbaren Vortrag. Noch ein kurzer Hinweis. Alle Videos und Unterlagen der Preisträger sind in Kürze auf der Webseite der Jung-Stiftung zu finden. Ja, das war's. Das ist das erste digitale Ernst Jung-Symposium für medizinische Forschung 2021. Ich hoffe sehr, es hat Ihnen gefallen. Wir bedanken uns bei allen Beteiligten, ganz besonders bei Ihnen, Professor Büchel und Professor Brünung, dass Sie uns so professionell und interessant durch diese Vorträge geführt haben. Wir hoffen natürlich alle, dass wir im nächsten Jahr alle gemeinsam wieder unsere Gäste persönlich auf dem roten Teppich der Jung-Stiftung begrüßen können. Und vielen Dank für Ihre Aufmerksamkeit und bleiben Sie gesund.